Okay.
Yeah, great. So now we're live. We have to be careful what we say.
Okay, now I can leave for good.
All righty, take care.
Eric is going to be the new host.
All right, Christian, take care.
I'm going to listen on YouTube.
Okay, very good. See ya. We've all had to get too good at Zoom.
Yeah, exactly.
Better
5 minutes, all set.
Better than we ever imagined. How are things with the pandemic in Bordeaux?
Well, it's not. Yeah, it's ramping up again. I don't know how it's in the US, but France is not. I mean, Bordeaux specifically is okay, but yeah, in France in general, and yeah, we are organizing a chorale training, a French chorale course, and it's challenging to make sure nobody gets infected and there's no clusters. So yeah, it's challenging.
Yeah, I mean, things in general are ramping up again in most of the northern states in the United States. New York City itself is doing well, but we're definitely preparing for to have more cases than usual. Almost all the cases are people who are not vaccinated. Almost all the serious cases, I should say.
Yes, serious cases. Yeah.
Yeah.
Yeah, the good thing is so far the hospitals are not overbooked. So, and
good.
But couple of hospitals in Paris started to reschedule some other interventions. So that's starting to not smell good. So we'll see.
And I've heard some hospitals in Germany and Switzerland and Austria also have been having volume issues, difficulty accommodating everybody.
Yeah. Well, so COVID cases are accommodated in France. It's just that, they reschedule non-emergent, non-urgent interventions.
Got it. Yeah. So we'll just wait another couple of minutes, give people a bit more time to join in.
Yeah, we are preparing to travel less for the next 3-4 months.
Have you reinitiated business travel?
Well, I did a few of them. I went to Vienna for the Fence Cavalier Network, and I went to the Lateral Foundation, which is in the south of France, for the optogenetic meeting. But there were very small meetings. It was max 30 people. And now we are organizing this chorale training. So yeah, speakers are coming. Americans stayed on the safe side and did not come, which I think was reasonable.
There's still been a lot of hesitancy for people. Yeah, let me just tell everybody, it's just noon now, East Coast time in the United States. And we're just going to give people another minute to join in, and then we'll get started. Yeah, there's been a lot of hesitancy and concern for people, although it's certainly more normal this year than it was this time last year. Christmas last year was still pretty tough.
Well, hopefully we can have a nice Christmas break.
Oh boy, I hope so. Yes, absolutely. The world needs it.
Yeah, I'm getting my 3rd dose soon, so.
Good. Yeah, I got my 3rd dose.
Oh, good.
Yeah. All right, listen, it's 12:01, so I think we should get started. It's my great pleasure to welcome you all to this week's Worldwide Neuroscience Seminar, today being given by Anna Beyeler. And it is my pleasure to introduce my co-host today, David Haggerty, who's a graduate student at Indiana University. Before turning things over to David, I do want to advertise that our next speaker will be Christian Luscher, who will be speaking 2 weeks from today on December 16th. But for today, we have Anna speaking, and David, take it away.
Hi, everyone. Just a quick introduction. So Dr. Beyeler earned her PhD in neuroscience under Drs. Didier Leray and John Simmers at the University of Bordeaux. After receiving her PhD, she came over to the States and did her postdoctoral fellowship with Dr. Kay Tai at MIT, where she studied the role of amygdala circuits in anxiety and valence encoding. In 2017, she traveled back to France to the University of Bordeaux, where she is currently an INSERM-funded principal investigator studying the pathophysiology of insular circuitry and emotional valence states, amongst many other things, awards, diversity, inclusion, service. She's a very bright and interesting young investigator. And today she'll be giving a talk titled Linking Valence and Anxiety in a Mouse Insula Amygdala Circuit. So without further ado, you can take it away. And if you would like to hold your questions towards the end, you can put them in the Q&A, and we'll have some moderation at the end.
All right, so thank you very much, David, for this kind of introduction. You can see my screen okay?
Yeah, that's perfect, Anna. Thank you.
Excellent, excellent. So yeah, I'm very glad to be here today to present some of our recent work on the neurobiological link between emotional valence and anxiety within the insular cortex. So before getting into the data, I'll give a little bit of background and what motivates our work. So anxiety disorder today are still the first psychiatric condition with the highest prevalence, and in 2015 the Global Burden Disease Consortium has estimated that 17% of adult population worldwide was affected with anxiety disorders. There's also been reported that there is a sexual dimorphism in these disorders, and 23% of the women and 14% of the men are affected. And the reason for this dimorphism is still unknown. There are, of course, therapies available, but they remain limited because of their efficacy limits, but also because they have side effects. And the first step to develop better treatments is to understand the neurobiological substrate of these disorders. So among the different symptoms that the patients with anxiety disorder show, the main symptom, or the core symptom, is excessive worrying, but it has also recently been described that patients with anxiety disorder show a bias towards negative valence. So for example, they will assign more negative valence to neutral stimulation compared to a healthy population. So these psychological studies, together with human imaging, has led to the hypothesis that dysfunction of circuits encoding emotional valence are causally involved in anxiety disorders. So where to start, which circuit to look at? So there's an interesting meta-analysis that integrates the work from dozens of studies that has shown that two brain regions are hyperactive in response to negative valence in patients with anxiety disorders, and these are the amygdala and the insular cortex. So this suggests that these two brain regions that are highly interconnected contribute to the physiopathology of anxiety disorders. So the insular cortex in a healthy condition responds to stimulation of negative valence to a greater extent compared to neutral stimulation. And interestingly, when you present a stimulus of positive valence, the response will be lesser compared to a neutral stimulation. And what's even more interesting is that in different types of anxiety disorders, here I'm showing for the social anxiety, you have an overactivation in response to negative valence, but you have a switch of response to positive valence. So in these patients, the insular cortex is now starting to be active in response to positive cues. So this again suggests that the insular cortex is involved in or is dysregulated and potentially causally involved in the physiopathology of anxiety. So the insular cortex has been well described in humans, where it's located at the intersection along the Sylvian fissure, at the intersection of the anterior, of the frontal, temporal, and parietal lobe. And it has also been described in rodents, where it has a similar cytoarchitecture as in humans. So, and it's composed of agranular, dysgranular, and granular cortices. So it has been involved in multiple functions, including proprioception and pain, taste perception, conditioned taste aversion, valence, as I just showed you, anxiety disorders, as I also mentioned, and also addiction. So interestingly, it has also been shown, especially in rodents, that the insular cortex is functionally divided in an anterior and a posterior section. For example, a study from the Zucker lab has shown that if you optogenetically activate by expressing channel rhodopsin in the anterior insular cortex, you will induce a preference for this side of the stimulation. And so this suggests that activity in this neural population will induce positive behavior, positive valence behaviors. Conversely, when they activated the posterior insula, they found that the animal will avoid the stimulated space, suggesting that this region is involved in negative valence. So anxiety has also been studied in the posterior insular cortex, and the lab of Nadine Dugola in Germany has performed fiber photometry recordings using GCAM6 that they expressed in the glutamatergic neurons of the posterior insula, and then recorded the animals while they were in the elevated plasmage, which is the classical anxiety assay. And they found that when the mice were located in the closed arm, the activity was higher compared to the open arm. So there was a decrease of activity when the animal were located in an anxiogenic zone or an exposed area. They also performed optogenetic activation using channel rhodopsin, and when they activated these neurons, they found that the mice would spend less time in the open arm in the angiogenic space. So in conclusion, they found that the neurons of the excitatory neurons of the posterior insula are more active in safe spaces, and that activation of these neurons is anxiogenic. However, the role and activity of the anterior insula in anxiety-related behaviors remained untested. So for this reason, Sebastian, a former postdoc in the lab, decided to record the activity both in anterior or posterior insular cortex. So he expressed GCAM6F in excitatory neurons, and then made the mice perform valence assays, including sucrose consumption or quinine consumption. Quinine is a bitter substance, which is very aversive to mice. And then tested the animals in anxiety-related behavior tests, including the elevated plasmase and the open field test. So this is just an example of the location of the optic fiber within the insular cortex and the expression of neurons under the optic fiber. So when we recorded the animals, we had a LED that would turn on every time the mouse would lick the sucrose solution, which allowed us to synchronize the activity, the neural activity, to this licking event. And we found that in the 7 animals were recorded in the anterior insular cortex and the 10 animals in the posterior insular cortex, there was no significant difference in the activity between the baseline and the post-lick period. However, when we recorded the activity in response to licking the bitter substance quinine, we found that the posterior insular cortex had a significant increase of activity right after licking the solution. So if we summarize, we found that the excitatory neurons of the posterior insula are significantly activated in response to an aversive stimulation, which is consistent with previous reports, including from the Zucker lab. So, but what we were really interested about is the activity in response to anxiety in related behaviors. So here you can see a mouse which is located in the closed arm of the elevated plasmase, and here you have the open arm. And you can see the simultaneous recording of the calcium signal. And then you can see that when the mouse is entering the open arm, you have an increase of the signal, and as well as an increase of the number of transients. And then when the animal will return to the closed arm, you can see the signal decreases back to baseline. So we were very excited about this observation, but this is just 40 seconds of recording. So we averaged the activity across the 15 minutes of the session of this mouse, and we found the strongest activity in the open arm. We then performed this analysis among 13 animals and found that there was indeed a significant increased activity in the open arm compared to the closed arm. Interestingly, this was specific of the anterior insular cortex, as this was not seen in the posterior insular cortex. And then when we looked at the open field test, we also saw an increased activity in the anterior insula, but did not see a difference for the posterior insula. So if the anterior insula is encoding anxiety-related information, we hypothesized that depending on the position of the mouse within the open arm, the signal or the neural activity would be regulated with increased activity towards the end of the open arm. So we kind of see that trend, but we also thought that depending on the direction of the animal, if the animal is actually going out or back into towards the closed arm, the activity might encode different information. So we decided to plot these activity depending on the directionality of the animal, and found that when the animal is going out to explore the open arm, there is a significant increase of the activity in the last part of the open arm, strongly suggesting that these neurons are encoding anxiety-related informations. So yeah, this is calcium imaging. So what are the neurons really doing and or encoding during behavior? So we are currently starting to do electrophysiological recording. So Yoni, a PhD in the lab, just started these experiments. So this is very preliminary. It's basically his first mouse, but I still wanted to share with you. So here is the silicon probe he implanted in the anterior insula, and this is the result for the open field test. You can see the track of the mouse, and he recorded 16 units in this mouse and found a significant increase of activity when the mouse was in the center of the open field test. So this is very preliminary, and we're gonna record more animals, but we were excited to see this result, which suggests that the increase of calcium signal we're seeing is related to spiking activity. So we then decided to test the causal role of this activity on anxiety behavior. So Celine, current postdoc in the lab, decided to do these experiments, and she expressed the inhibitory thread or the control virus in the excitatory neurons of the anterior insula. You have the expression here. And then 4 weeks after viral expression, she injected CNO 30 minutes before the elevated plasmase, and the successive day repeated the procedure before the open field test. And she found that when you inhibit the excitatory neurons of the anterior insula, you actually increase the time in the open arm in the second part of the test compared to the control group. And we found a similar increased time in the center for the open field test. And this was not due to changes of distance travel or speed of locomotion. So in conclusion, we found that the neurons in the anterior insula increase their activity in the angiogenic spaces of the elevated plasmase and OFT, and that they play a causal role, because when you inhibit these neurons, you decrease the level of anxiety of the mice. So of course, our next question was like, which circuits are supporting this increased activation and this control over anxiety-related behaviors? So as I mentioned in the introduction, the amygdala is another important region that is dysregulated in patients with anxiety disorders. It is known to respond to stimulation of negative valence from different fMRI studies, but it has also been shown that it will be activated in response to pleasant stimuli, even more to a greater extent compared to negative stimulation. Studies in different animal models, including mice, has shown that the amygdala, specifically the basolateral amygdala or BLA, is necessary for learning association of negative and positive valence. And while I was at MIT, together with Ada and Praneeth in the Thai lab, while it was still at MIT, we have found that neurons within the BLA respond to positive and negative valence, and that these neurons are intermingled, although they display a dorsal ventral gradient from negative to positive valence. We also found that among the wide range of projection targets, the neurons projecting from the BLA to the ventral hippocampus have angiogenic properties, that the neurons projecting to the nucleus accumbens encode positive valence, and the neurons projecting to the central amygdala encode negative valence. So I hope this at least this convinced us that BLA was an interesting target to study the output from the insular cortex to the amygdala. But before starting our study, we wanted to characterize the output pathways of the insular cortex. So we used a simple anterograde barrel tracing approach by expressing EYFP under a ChemK2 promoter in the anterior and posterior insular cortex, slice the brain, and perform confocal imaging in 12 downstream regions of interest. So I'm not going to go in detail of all brain regions, but what you can see from this example animal of the anterior insula is that the main projection target is the BLA, but you also have some projection in the central amygdala, and specific is mainly the lateral part, and that the second main target is actually the contralateral anterior insular cortex, but not the posterior one. And if you look at this example of an animal injected in the posterior insular cortex, we clearly see that the main projection is the central lateral amygdala and also the central medial, with some projection to the BLA. And interestingly, we found that the contralateral posterior insular cortex also receives dense projection. So this is the result for the 8 animals we quantified. I'm not going to go in details of all these brain structures, but here is the summary of this data, where the width of each line represents the density of projection. So you can see that indeed the anterior insular cortex mainly projects to the BLA, but still has substantial projection to the medial and lateral part of the central amygdala. And interestingly, the contralateral anterior insula also received dense projection, but not the posterior. The posterior insular cortex, conversely, mainly projects to the central amygdala, but you also have important projection to the basolateral amygdala. And you have a specificity to the contralateral posterior insular cortex. So this really strong difference between the projection to the contralateral side is important to us, because it's kind of a control that the strong projection that we see to both BLA and CEM from the anterior and posterior insula is not an artifact from injection that would not be performed correctly, and it's really due to the projection pattern of the neurons. So based on previous work, including ours, we decided to focus on the BLA and CEM projection from the anterior insula. And here you have an example of neurons of the anterior insula that are projecting to the central amygdala. So this is just a qualitative approach to look at these neurons in the whole brain. But Anis, former postdoc in the lab, decided to quantify the synaptic properties and intrinsic properties of the neurons of the anterior insula projecting to the insular cortex overall, projecting to the BLA and the central amygdala. So she injected a viral vector expressing channel rhodopsin in excitatory neurons, and then recorded postsynaptically either in the BLA or the central amygdala. And with this approach, when she shined light over the recorded neuron, she could induce excitatory postsynaptic current. And when she applied sodium and potassium channel blockers, she still had a current, meaning that there was a monosynaptic component to the current that was abolished by glutamatergic antagonist, showing that these currents were indeed glutamatergic. So this is the quantification over all the cells she recorded. So there was monosynaptic projection from the insular cortex to BLA neurons as well as CEM neurons. And she also found that when you activate these terminals within the BLA or CEM, you induce an inhibitory current, which is blocked by TTXN4AP, meaning it's a polysynaptic response. And yeah, see it. So here, sorry, it's quantified here where you see that the current is abolished by the network activity blockers. So she also studied the dynamics of these currents, of these synaptic inputs, sorry. And you can see here that you have a depressive kinetics that is present for both the inputs to the BLA and the central medial amygdala. And interestingly, the depression is stronger for the inputs to the BLA compared to the CEM. She also quantified this kinetics over on the inhibitory currents and found that it was present for both synaptic inputs and also stronger for the inputs to the BLA. So she also wanted to quantify the recurrency of connection between the anterior insula and the BLA. So she co-injected a viral vector expressing channel rhodopsin EYFP and a retrograde tracer CTB555 within the BLA. And then she recorded within the anterior insular cortex. And she found that the neurons that from the AIC that project to the BLA indeed receive monosynaptic input back from the BLA. She also tested this within the BLA. So in that case, she co-injected the viral vector and retrograde tracer in the anterior insular cortex and recorded in the BLA, and found that the neurons of the BLA that project to the anterior insula receive monosynaptic inputs from the anterior insular cortex. So really demonstrating that you have direct monosynaptic loop between these two regions. So this doesn't quantify the amount or the proportion of this direct recurrent loop, but shows that it exists. So finally, she characterized the intrinsic properties of the neurons of the insular cortex projecting to the BLA or the central medial amygdala. And here you have example of the injection of retrograde tracer she did in the BLA or in the central medial amygdala. She then recorded within the insular cortex the neurons projecting to the CM or to the BLA. And here's just an example of neuron that we were filling with biocytin to confirm the co-staining with the retrograde tracer and biocytin. So she recorded a lot of electric properties. I'm just going to show the one that were different. So here you have the membrane capacitance, and you can see that it's smaller in the insula CEM neurons, suggesting that these neurons are smaller than the neurons projecting to the BLA. She also found that the firing threshold is slightly lower for the neurons projecting to the CEM, suggesting they're more excitable. And when she did the input-output curve and quantified the firing frequency depending on the current injected, she found that the neurons projecting to the CM were indeed more excitable for low current injected, but when she injected higher currents, it was actually the neurons projecting to the BLA that would start to fire with a higher frequency. So this was very interesting and suggesting that these two pathways projecting from the insular cortex to the BLA and the CM have divergent electrophysiological properties within the insula, but also at the synaptic terminals in the downstream target. So after characterizing these different pathways, we decided to first focus on the projection from the anterior insula to the BLA to test their functional role. So if we go back to our conclusion diagram of the activity of the anterior and posterior insula, we hypothesized that the projection from the anterior insula to the basolateral amygdala are responsible of the increased activity in the elevated postmaze and open field tests. So this has been a team effort led by Céline Nicolas, the postdoc, together with Ivan and Azim, that were two master students in the lab. So first, they wanted to test the causal role of this projection in anxiety and valence-related behaviors. So we decided to take advantage of a very new tool, bipoles, which has recently been published by the lab of Simon Wiegert in Hamburg. And it's a new tool which allows bidirectional, because it's a bidirectional pair of opsin for light-induced excitation and silencing. So it's composed of two opsin, one inhibitory opsin, GTACR2, and one excitatory opsin, crimson. So with this protocol, you can induce inhibitory currents by applying blue light to the neurons, and excitatory currents by applying red light to the neurons. So this is a trace from the Nature Communication paper I just cited. So they injected mice bilaterally with viral vector expressing as a somatic bipole. So these opsins were targeted to the soma through a protein from the potassium channel. So it would link the opsins to the cell body. And then, so they used a dual viral approach by injecting a retrograde CAF2 virus within the BLA and the anterograde virus expressing the some bipole in a Cree-dependent manner. And so here is an example of the neurons expressing this bipole tool and the optic fiber placement in the insular, just above the insular cortex that allows the activation and inhibition of these neurons. So we used a protocol that was established before. So to inhibit the neurons, we used 5 millisecond pulses at 20 hertz, about at 20 milliwatt. And to activate the neurons, we used 20 millisecond pulses at 20 hertz for at 10 milliwatt. So this has been published before, and decided to use these. So we hypothesized that if you activate or inhibit these AIC to BLA neurons, you would have nice bidirectional control over the behavior. However, that's not exactly what we observed, and we found that overall, in the group that was where the activity of the anterior insula to BLA neurons was manipulated, the neurons would spend less time in the open arm, but it was not dependent on the activation or inhibition of the neurons. And when we did the open field test, we found that this trend in the EPM was significant and that the animal would spend less time in the center of the open field test. So there are different interpretation for this, and I'm happy to discuss them after my presentation. But one of them is that if you alter the activity of these neurons, you will increase the level of anxiety of the mouse. And we also checked for the distance traveled by the animals and did not see a difference when the neurons were activated or inhibited. So the impact on the anxiety seems to be specific of this behavior. So in conclusion, altering the activity of the AIC to BLA neurons increases anxiety-related behaviors. So as I mentioned, we found that both activation or inhibition did not have a time-locked influence on the behavior. So we wanted to test whether we were really activating the neurons with our protocol. So before sacrificing the mice, we activated the neurons with red light, and after did immunostaining for the CFOS, marker of neural activity. And we found that indeed, the CFOS expression, proportion of neurons expressing CFOS was higher in the neurons expressing the some bipole opsins. So because this manipulating experiment was not so conclusive, we decided to record the activity of these neurons and try to understand how they encode valence and anxiety information. So we used the same dual viral approach, but this time we expressed the GCAM6M. And here you have an example of a mouse expressing the sensor and the optic fiber implanted in the insular cortex. So we tested anxiety-related behaviors, the EPM and OFT, as well as Valence, including sucrose consumption and mild electric food shock. So for the elevated plasmase, here is again the heat map of the activity, the neural activity for 20 mice. And you can see that the activity is stronger in the open arm compared to the closed arm. And the analysis showed that this was significant. So confirming that, or hypothesis that the increased activity of the entire insular cortex might be supported by the projection neurons of the anterior insula to the BLA. In the open field test, we also found an increased activity in the center of the open field test. And in these experiments, we also quantified the transient activity. So here you have an example of the global signal. So here I zoom in on the gray area, and then we filtered the signal and applied a threshold to be able to detect transients that would reflect bursting activity of the neurons. And when we quantified this in the elevated plasmase, we found that there was a trend for an increase activity, an increase of number of transients in the open arm, and we found a similar trend in the open field test. So this was not very strong effects. However, when we quantified the difference of frequency in the open and closed arm, and we correlated that with the time spent in the open arm, which is inversely proportional to the level of trait anxiety of the animal, we found that it was a positive correlation with trait anxiety. So this means that the more different the signal or the frequency of transient between the open and closed arm is, the more anxious the animal is. So in conclusion, this analysis really links the neural activity of the AIC-BLA neurons, not only with state anxiety, but also with trait anxiety. So in conclusion, for these experiments, we found that if you alter the activity of the AIC to BLA neurons, you increase anxiety-related behaviors, and that the activity of these neurons is increased in anxiogenic spaces in both tests, both tests that we used. So the AIC-BLA neurons indeed are more active in the anxiogenic spaces, and now we wanted to assess the role of these neurons in valence. So we used a more classic optogenetic approach by expressing channel rhodopsin to activate the neurons, still using the dual viral approach. And here you have an example of the expression and fiber placement in a mouse. And for inhibition, we used GTACR2, which is the modified channel rhodopsin that will be activated by blue light and then inhibit the cell as it is permeable to chloride ions, with the same dual viral approach. And here's an example of the expression and the fiber placement. So for the optogenetic activation, we found a trend to an increase, but this was not significant. So the mice tend to spend more time in the stimulated zone, but it was not conclusive. Before using the GTACR, we decided to test this tool because it was a new tool for us. So we recorded neurons expressing GTACR, and you can see here that if we triggered firing with a current step, you could induce the firing that was completely blocked by a pulse of blue light. And we also tested different stimulation or inhibition protocols. And you can see that if you use 20 Hertz with 5 millisecond pulse, you also completely block the firing of these neurons, which is due to the properties of the opsin and the time constant of the opsin. So this is interesting because you can use exactly the same protocol to activate and inhibit the neurons. And this means that you can use the same control group between activation and inhibition. So when we inhibited these neurons, we were surprised to find that the animals were indeed preferring the site of stimulation. which was not expected, as previous literature had found that the activity or activation of these neurons would induce a preference. So in our case, the inhibition of the AIC-BLA neurons induced a place preference. So this suggests that they are encoding negative valence. So to test this, we recorded the activity of these neurons in the Valence, different Valence tests. So this is again the sucrose quinine test. And you can see that when the animal was consuming the quinine, so this is the lick onset, the activity of the neurons would decrease significantly. However, when we recorded during the consumption of quinine, we did not see a difference, although we were expecting an increase of activity in response to negative valence. So the AIC-BLA neurons are inhibited by a positive valence, but in this gustatory valence task, we did not see a difference for the quinine consumption. So we decided to test other stimulation of negative valence. And first we did not fear conditioning, actually just a mild food shock that was delivered to the animal. And in that case, we saw a strong increase of activity of the AIC-BLA neurons. We also did a second aversive stimulation to the mice by tail suspending them. And we also found an increase of activity after this stimulation, showing that these neurons are active in response to negative stimuli. So if we go back to our conclusion slide, we found that the neurons of the AIC-BLA bidirectionally code for negative valence. So as I showed you earlier, the anterior insula is projecting to multiple downstream region, and we were particularly interested in another projection target, which is the lateral hypothalamus, and specifically the rostral part, as it has been shown to relay information from the insular cortex towards downstream projection that control heart rate, and which is a physiological response of anxiety. So to test whether the activity of the AIC-BLA neurons is specific for this population, or if all the neurons of the anterior insula or all excitatory neurons of the anterior insula respond in a similar way, we decided to record in this neural population that potentially encodes different information. So this is the work of Anna Marta, and it's still preliminary, who's was a master's student in the lab and is now a PhD student in Paris. So she expressed GCAMP6 in the anterior insular neuron that project to the rostral lateral hypothalamus, and in other set of experiment expressed channel rhodopsin in these neurons, and then tested them in valence and anxiety assays. So for the elevated plus maze, so here you have the average heat map of all the animals you recorded, and you can see that clearly the pattern is different from what we observed for the AIC-BLA neurons. And it was not significant, but it's a strong trend for a decrease of activity in the open arm. So, but clearly it is different from the other population we recorded from. For the optogenetic manipulation in the elevated plus maze, she used off-on-off protocol with 3-minute bin, and computed the ratio of time spent in the open arm during the off and on period, and found a trend to more time spent in the open arm when we activated these neurons. For the open field test, in that case, there was a significant increase of this ratio of time spent in the center. So activating these neurons will increase the time the animal will spend in the angiogenic zone. And finally, for the real-time place preference, she found a trend to a preference for this the stimulated side compared to the non-stimulated zone, suggesting a role more in positive valence. So now I'm gonna share another very preliminary results. So we started to actually record these two populations together. So the neurons that project to the BLA and the lateral hypothalamus, just to confirm whether they indeed differentially encode valence and anxiety-related information. So this is a little more complex experimental design. So this is a quadruple viral strategy based on Cree-dependent expression of GR gecko, which is a red calcium sensor, and the flip-dependent expression of GCAM6F. So she injected the retrograde CAV2 Cree in the BLA and the retro AAV flip vector in the LH, and then a one-to-one mix of these two anterograde Cree and flip-dependent viruses within the anterior insula. Here you have an example of the expression of these two calcium sensors within the insular cortex with the optic fiber placement right above. And then she recorded the activity of these neurons in the elevated plus maze. And then here you can see the simultaneous recording of these two neural populations. And you can see that in the open arm, you have transients in the AIC to BLA population. And you also have activity in the AIC LH neuron. But then when the animal goes to the closed arm, you have a big increase in this AIC LH population, which is stronger compared to the AIC BLA population. So this is still very preliminary. We are performing more experiments and more recording to confirm whether these two activity are divergent when you record them simultaneously. So in conclusion, for this preliminary set of experiments, we found that in the elevated plus maze, the activity of the AIC LH neuron tend to decrease. We are still analyzing the data for the open field test, and we found that it is increasing the time spent during the optogenetic stimulation, so suggesting a role in positive valence. So although I don't want to make big claims about this experiment, but I think what we can take of that experiment is that there are different populations in the anterior insular cortex that will encode information relative to anxiety and valence in a divergent way. So before going on, one of the important aspects when you study neural circuits is to try to identify molecular markers of these projection-defined neurons. So you can use a more comprehensive approach by doing RNA sequencing or single-cell RNA sequencing, for example. But we decided to do a more targeted approach, and we based our hypothesis on the fact that SSRIs are the first-line treatment for anxiety disorders, and that the serotonin 1A receptor is involved in anxiety disorders, as agonists have anxiolytic effect in patients as well as animal models, and the serotonin 2A receptor also regulates anxiety levels. So we hypothesized that maybe this serotonin 1A and 2A receptor would be differentially expressed in the insular cortex and in specific neurons projecting to downstream region. So these receptors were known to be expressed widely in the brain, mainly in cortical regions, as well as in the insular cortex. However, it remained unknown in which neural population these receptors are expressed. So Anes did this experiment while she was in the lab and quantified the expression of the 1A or 2A receptor using immunostaining in glutamatergic neurons. So here you have mu-N staining, and here you can see the serotonin 1A receptor, as well as in GABAergic neurons. So she did both mu-N and GAD67 staining, and you can see here that there is expression in this inhibitory neuron. She also used retrograde tracers to target 4 different projection neurons. So the neurons projecting to the VLA or the central medial amygdala, the neurons projecting to the rostral or caudal lateral hypothalamus. And so in summary, she found 2 interesting things. So the first one is that most of the excitatory neurons, actually 80% of them, expresses the 1A receptor, whereas only 30% of the inhibitory neurons express the 1A receptor. She found very similar proportion for the 2A receptors. So meaning these 2 receptors are differentially expressed in a molecular defined population, depending on the whether the neuron is an excitatory or an inhibitory neuron. So then regarding our hypothesis of differential expression, depending on the projection target of the neuron, she actually found that regardless of the projection, both 1A and 2A receptor were highly expressed in these different neural population. So there was no differential expression of the 1A or 2A receptor, depending on where the neurons are projecting. So with that, I'm gonna summarize what I showed you today. So I started by showing you that the excitatory neuron of the anterior insula have anxiogenic properties using recordings with calcium imaging and manipulation with DREADS. I showed you that the AIC-BLA neurons have an anxiogenic role. So we did recordings to see that they have a higher activity in anxiogenic spaces, and with optogenetic manipulation, we increased anxiety-related behaviors. And interestingly, the activity of these neurons is at least different, and it seems to be opposite to the activity of the AIC to LH neurons. And finally, we found that the AIC neurons projecting to the VLA code for negative valence, and that projection neurons of the anterior and posterior insula uniformly express the 1A and 2A receptor. So what are the next steps after that? So as I showed you, we started doing single neuron recordings, and we are going to pursue that to really dissect what the coding properties of these specific circuit in the insular cortex based on their projection, but also based on molecular properties, so different types of receptor expression. We are also investigating the synaptic plasticity in these different output pathways, depending on the level of anxiety of the mouse. And finally, of course, the holy grail is to study this different neural population in pathological condition, including anxiety and addiction. So stay tuned for more work on that. So with that, I would like to thank the entire lab, the current and past members, as well as our collaborators and our funding sources, and of course, you for your attention. And I'm happy to take any questions. Thank you.
Fantastic, Anna. Thank you so much for a great talk. I do want to remind our audience to please put questions in the Q&A function. In the meantime, David, I'll turn things over to you if you want to get things started.
Okay. Well, we have one question so far. It says, hello, great talk. In the bipole experiment, have you tried to reverse the order of activation and inhibition?
Yeah, that's actually an excellent question. And looking back at that experiment, we didn't choose the right order, I think, because especially when you do inhibition, you might have a rebound. And so we decided to do activation and then inhibition and then nothing. So actually the nothing might be contaminated by this rebound activity. And whether if you start with the inhibition, then if you have a rebound and then you have activation behind, this would be integrated together. So yeah, clearly when we are going to repeat that experiment, we're going to start with the inhibition and then do the excitation. And yeah, So this is still, there's a lot of more things to do with this tool. It's really an exciting new tool, but yeah, there are these type of things that you don't always realize when starting these experiments. So hopefully it helps others that want to use it. I think it's available on AdGene now.
Mm-hmm. So we have a question from Mengyu Liu. Interesting talk. As you mentioned, anxiety is sexually dimorphic. I am wondering if you observed any sexual dimorphism in anxiety-related behavioral assays or neural recordings.
So yeah, that's a very good question, and it's also one of our interests. So we perform all our recordings, both in male and female. I didn't mention it, but it was always written. And of course, we compared. So in terms of behavior, it's pretty similar. We just observed that the variability is higher in male than in females, at the contrary of what was expected from.
Yeah, that's interesting.
Yeah, but it has actually, so after going back to the literature, this has actually been published already. And even though they have the cycle and we didn't track for the cycle, they are less variable than males, which and the variability in the male is likely due to the rank, the social rank of the animal, and probably testosterone levels. But yeah, so they're more variable, but there are similar levels. So the time spent in the anxiogenic and safe zone is similar in the male and female. And regarding the neural activity, so our sample size is still, about like 9-10 mice for each group. So male plus female, but so we did not see difference. So I think we would need a little more recordings to make a absolute statement, but so far we did not see differences in male and female. So there might be other mechanism, and yeah, there are also mice, so we don't know how much this difference or this sexual dimorphism that we observe in human exists in mice. And I think actually it's a very interesting model because there is all these social construction that humans have and mice don't have. So I think the fact that we did not observe this difference might say something about the neurobiology also.
Cool, great, thank you. Dave, go ahead.
Next question we have is, were you able to record neural activity of insular neurons during the activation inhibition with the blood pulse?
Yeah, no, we did not do that. We did some slice recording, and this has been also characterized by the lab of Simon Bigert. So you can activate and inhibit the neurons. Like in vivo, they did experiments with pupil dilation, but yeah, we did not record, and I think that would be really nice. And one of the possibility is that we actually don't inhibit as much, and that the previous activation is still continuing during the inhibition phase. So it would be really interesting to record these neurons while we are manipulating them. But that starts to be a more challenging experiment.
From Zijun Wang, hello, great talk. For the optogenetic iPSC, do you think it is the direct GABA projection from insular cortex to amygdala, or inhibitory feed-forward local circuit within amygdala?
Yeah, great question. This is definitely a local circuit because there is no long-range projection from the insular cortex to the amygdala. There is no long-range inhibitory projection, sorry. So we have done these experiments using transgenic mice, so VGAT mice, and doing retrograde tracers from the BLA, and we did not see any co-labeling. And we also did anterograde tracing and did not see projection outside of the insular cortex from the inhibitory neurons. And then when we block the network activity within the BLA or the central, we completely abolish the inhibitory response. So it's clearly local polysynaptic feed-forward or feedback inhibition. This we don't know, but yeah, it's clearly local.
Great. We have amazing talk, super cool work. What do you think are the implications of the inverse role of the anterior insula to the lateral, to the LH projection, especially since the initial hypothesis was that they could serve as the effector of physiological change for anxiety? Also, another question, are you planning to look at the AIC to CEM role in anxiety?
Well, a lot of great questions. So I'll start responding to the second part, and is yes, we are actually currently performing the dual recording, what I showed you for the AIC-BLA and AIC-LH. So we're currently doing that for AIC-BLA and AIC-CM. With the hypothesis that the activity would be similar, but yeah, so I cannot speak to that. The experiment are actually running like these days, so mice are still alive. So regarding the different response and of the AIC-BLA and AIC-LH and the effector role, so it's not very clear yet which response the AIC to LH physiologically induce. So whether it would be an increase or decrease of heart rate, or if it has an impact on other physiological responses such as breathing. So this is one of our planned experiment to actually activate or inhibit this AIC to LH projection and record the heart rate to see whether it would be more an anxiogenic or anxiolytic component, physiological component.
Okay, fantastic. So we do have time for one last question. This is from Yanis Bakagianis. Regarding serotonin 5-HT2C receptors are also known to be implicated in anxiety as well as other receptors. What is your hypothesis regarding other receptors?
Well, yeah, I mean, that's a great question. And yeah, so my hypothesis would be that they are also uniformly expressed in the different projection neuron. But yeah, we decided to actually change strategy and go for the comprehensive approach with RNA sequencing. And one of our hypotheses is actually rather than just the presence or absence of these serotonin receptor, the balance or the proportion of these receptor could actually tune the activity of the neurons, as the 1A inhibitory receptor and the 2A excitatory receptor. This could be a finer tuning level of the activity. So yeah, that would be my hypothesis. That still needs to be tested. Well, you're welcome to come to the lab and help us do that.
Absolutely. So thank you very much. And I want to thank you for a wonderful talk today. I want to thank David for being my co-host. David, good luck with your own PhD work. And thank you all for joining us today. Just a reminder, we do have our next Worldwide Neuroscience talk scheduled for 2 weeks from today, December 16, and the speaker will be Christian Luescher. Thank you, everyone. Have a good couple of weeks.
Thank you. Bye-bye.
Thanks again, Anna. Bye-bye.
Bye.
Bye.